part, we are going to kind of talk about some minor things about kind of brain physiology. I'm sure you all know about kind of the neurons, the structure of neurons, how different parts of the neurons are interacting with each other. We have axons, we have dendrites, how these things are interacting, and we have synapses, which is connecting different neurons together, okay? We have what we call action potential and resting potential. So what we have basically, we have, if I record the membrane potential, if I just put an electrode inside the cell and I record based on T the voltage that I have inside the cell, and if it is 0, then I have a recording of something like minus 70 millivolt. So it is just a kind of really low level of negative voltage inside the cell. And then when we have action potential, what we have is that minus 70 will just go up. If we stimulate a neuron and increase the level of resting state potential to a certain degree, let's say minus 50, then the neuron would be firing, just going back. So this is called kind of the threshold that we have. And if we get neuron to that threshold, then it would be something like 0 or 1 response. So we have the action potential or we do not. So if we get to that level, we will have an action potential. We will be back to the normal situation. And this is the T, so it is over the time. So when we have action potential, we have a huge influx of positive ions into the cell, and that influx would make the kind of neuron potential to something like plus 40. So it is just going to increase, and this is what we call neural firing. And the interesting thing about this neural firing is that neural firing would just go into the kind of axons from here and just go with a really rapid kind of speed to the end of the axon. So that is kind of what we call the action potential that is propagated through the axon to the synapses. So that is how the system is basically working. I'm not going to the details about that. There are many technical details about the ion channels and how they are working together, how pumps are interacting. These are not the things that we are going to discuss in this class together. So we have the action potential here. We can have an action potential here, and then the action potential would just go ahead, and then this action potential would go to the end of the axon terminal, and then we will have synapses, and then the next neuron. So we will have the next neuron, and then the action potential, which is basically 0 or 1. So we go back. So as you can see here, then this 0, 1, 0, 1 would get to the kind of synapses and would go to the next neuron and stimulate the next neuron. So that is how a sort of a bioelectrical impulse would go into the axon and would go to the next neuron. And we technically can record that. We can just put an electrode and record that. This is exactly what we record, and we call that a spike. Okay, this is the resting potential. This is the spike, and this is, as you can see here, there is an increase in the resting membrane potential from something like minus 7 to minus 50, and as soon as we get to the threshold, then we will have a spike, and then the rest. Okay. And this is exactly what we do. We stimulate the neuron to that. This is basically 10 millivolt of stimulation that would make the neuron to fire and just go back to the previous situation that the neuron was inside. And when we have this process, there are some important basic principles. So if we have, let's say, neuron 1, neuron 2, and neuron 3 that are entering to the neuron 4, and this one is bringing an action potential, this one is bringing an action potential, this one is bringing action potential. How neuron 4 is responding to this aggregation of stimulation would be basically based on the firing that we have from here, and then for each synapses, there are some strong synapses that would make a significant impact on the next neuron. There are some kind of synapses that are, so some of them are positive, some of them could be negative. And then we have, let's say, we need 10 millivolts for the stimulation. This one is bringing one kind of positive, so let's say 2 millivolt. This one is bringing 7 millivolt. This one is bringing, let's say, 3. And then they would, because this is a negative, then at the end we will have something like 6, which is not enough to stimulate the next neuron. So we need to have enough stimulation from other parts to be able to stimulate the next neuron, okay? And the next neuron would be firing, and so that the response would go down. So this is something like a simple decision process. So in different parts of the brain, for example, we have cortical neurons that would go from the cortex to the limb to just do something with your limb. When they want to fire, they basically, we have different inputs to this system, and these inputs are gathered together to reach to a level to kind of turn on this kind of pathway to be able to have a movement in the muscle, okay? And based on the positive and negative inputs and how they are kind of aggregated to each other, then we will have a response at the end of the day. So we have kind of different inputs, excitatory neurons, negative or inhibitory neurons. At the end of the day, we will have an output from this system. So this is the kind of the way that this system is working. The other important part of this system is that some of these neurons that we are talking about, they can have spontaneous firing. So it is not the case that for having a firing, we need to have an input from a part of the system that would go to one part and then would make a specific kind of action to happen. Sometimes we have areas within the brain that are technically generating the action potentials, and those action potentials could make some specific activities. That is the reason that sometimes when we are, for example, in the REM sleep, when we are asleep, there are some spontaneous activity in thalamus that keep the cortex active without any input to the thalamus. So thalamus by itself could make some firing and just turn on some other parts of the cortex by the firing that it's making by itself. And it is amazing that it is not the case that all the neurons need inputs to make firing. Some of the neurons are able to make firing or make signals without receiving inputs. So they can make a spontaneous firing. And it's another interesting thing that why kind of these parts could make what we call kind of dreaming during the sleep. And I will discuss about those things in the next talks in terms of how these spontaneous firings are going to be important in terms of the different activities that we have in the brain. And we'll discuss about those things. The other important thing about these firings in the brain and these signals, these impulses in the brain, these impulses or pulses of electricity or bioelectricity in the brain, is that some of them could be in tonic or phasic. So, for example, here, at the end of the day, you can, for example, here you have, let's say, 20 impulses, okay? Here you have 20 impulses as well, but these are coupled together as we call phasic kind of firing. And sometimes they are in the same rate as we call a tonic firing, okay? And it's going to be important in terms of whether the neuron is firing in a tonic way or phasic phase. And these two kind of ways of firing would make different levels of, let's say, information. So they have different types of information inside. And it's amazing that how neurons could, with the same amount of firing, with different patterns of firing, cells in the brain could transform different sorts of, or transfer different sorts of information. So this is going to be critically important for you to understand that even the pattern of firing is going to be important in terms of the information that different parts of the brain are transferring. Okay, I have somebody who has the mic on and is clicking, and just have like the sound of clicking my. So if kind of the organizer could just help me to mute everybody, getting sure that clicks are not being heard here. Okay, good. Okay, so with that tonic, let me just give you one single example, interesting example that might be interesting for you. Okay, so when we give mouses alcohol, they learn over time that if they press this pedal, they would receive alcohol from here. So they learn, okay, they need to press this and then get alcohol. And we make them dependent to alcohol, okay? And then we make them dependent to alcohol and then put kind of these pieces that I told you, VTA and nucleus accumbens. We can just put a laser beam inside the brain and stimulate nucleus accumbens, okay? Or stimulate kind of VTA. And stimulate with a phasic firing or tonic firing. And if we do with the phasic, this will be the amount of dopamine that we will release. And this would be the amount of dopamine release at the end of the day that is released here as well. So the amount of dopamine release is the same, but this is the tonic release or tonic stimulation. This is the phasic stimulation. And when we do the phasic stimulation, it would make the rat to press the lever. But when we do the tonic stimulation, it doesn't press the lever. So the amount of dopamine is the same, but the response could be different. And that makes things even more complex. That's the reason that sometimes we give people the same amount of medication, but because we cannot give the medication in, let's say, tonic or phasic way, we cannot do the expected behavior as we are looking for. So it is quite amazing that how these even tonic and phasic patterns of neural firing are going to be important. This one is being published in March 2020 in a journal that is called iScience. So those who are interested to know about iScience, they can tell me that what is the impact factor of iScience that is being published by Cell Press. And by I, they mean interdisciplines, that interdisciplinary journals are publishing different things. So it's also important to know when it has been published and, I mean, where it has been published and what are the things about this journal. This journal is being online from 2018. So it's a kind of interesting journal, and they are publishing really high quality papers. Okay, so that is what we can have in terms of even the kind of the phasic versus tonic response are going to be important. I will tell you when we talk about reward system and decision making, how these tonic and phasic responses are going to be critically important. Can we do the same in the kind of living human brain? Yes, we can do. This is called the kind of deep brain stimulation and deep brain recording that it is technically doable. So it is one case that you can see that this is the. Could you tell me this electrode? Which one is this electrode? 1, 2, 3. Which one is this one? This is exactly the three-dimensional kind of view that you need to get. First of all, what is the cut? Is this the coronal or sagittal or axial?
Axial.
Coronal.
This one.
Is coronal.
This one is coronal, yeah. Coronal, right. What about this one?
Axial.
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 Okay. So this one is going to the kind of the transverse. So that is the electrode that you have. This is the electrode number 3. And you can see here the cut from the electrode. This is the tree, the number 3. As you can see, this is the direction. And this is the direction that we have with the... And when we cut that with the coronal, so if we cut that with the coronal cut, you can just see this cut, this really small cut. And do you know where it is? Do you know what is this part of the brain? These both sides? Any other suggestion?
Hippocampus.
Yeah, it is. As you can see here, you need to have the type 3-dimensional kind of view. So this is the temporal cortex, right? This is the temporal cortex. And we are talking about, this is the superior temporal gyrus, this is the middle temporal gyrus, inferior temporal gyrus. And then this one is the medial temporal gyrus, which is in the medial part. And we have, yes, we have hippocampus here, and we have many other areas here. But sometimes they call that medial or mesial temporal gyrus. And why they are putting the electrode in the medial temporal gyrus? Why here? Does anybody know that why they are putting the electrode here in this part of the brain?
For memory.
For memory improvement. So if I want to have a better memory, I can put the electrode here and just stimulate those areas.
Maybe for seizure improvement.
Now we have Khadija from Kerman.
Yeah. Because the neuron in hippocampus are sensitive to seizures, so deep brain stimulation in this way can be a very good way for improvement in seizures.
Perfect, yeah, that's true. That part of the brain is really sensitive to making seizures. And when we kind of put electrodes there, first of all, we can record that it is the source of seizure or not. And then those who have seizure, we can sometimes just stimulate that part of the brain to control the seizure. And if it is not going to work, they basically need to remove that part of the brain to control seizure. But with electrodes, as you can see here, we have different dots here. So these electrodes are recording from different parts of the brain. So based on which part is firing during the seizure attack, they can remove that part from the brain to just control that kind of really detrimental seizures that these people are experiencing because of the high activations in this area. So we can't control those things. In the next chapter, we will discuss about some of these deep brain stimulation potentials. We'll discuss about them later on. Okay, so that is the part about some kind of neurophysiology parts. And then we are going to the fourth part, which is going to be about some cognitive neuroscience and how these parts of the brain are working together.